So, welcome to another wonderful lecture in molecular biology. Yay, woohoo. Best subject matter ever, except for viruses for next term. Which reminds me, I know some people are thinking about courses to take next term. It seems like a way off in the future. But I will be teaching virology next term. I'm also going to be teaching what's known as Recombinant DNA Techniques Lab, which is 431. That's actually completely separate from the recombinant DNA techniques. So there's actually a theory and a lab. Basically, the reason that those two are separated from each other is that you cover what's done in the lab in about a week worth of lecture. The other thing, which I think is totally awesome about the lab, is that recombinant DNA techniques laboratory is horrible as far as the name is concerned. What it really is mutant viruses from hell, which is actually exactly what you would be working on in this lab. It's a course-based undergraduate research experience, and if you're more interested, come ask me afterwards or send me some emails about that particular course. I think it's great, but then again, I teach it, so I might not be the best person to ask about that. So today we're going to finish up with what people sort of call DNA repair, which I think is just horrible, and recombination. This is talking about jumping genes and the transposons that are jumping around in the genome. And then we'll move on to transcription in sort of the second half, last third, last quarter, whatever it is, of this particular lecture. And that is going to bring us to pretty close to on track with the syllabus. So I know a couple of people had questions about the syllabus and where we stand on that. So it should be getting us pretty close. So transposons. These are, I think, almost as cool as viruses. And in fact, some of them are really similar to viruses in terms of what they do to genomes. And last time we talked a little bit about mutations, and the time before, what the changes that happen to the DNA in terms of what kinds of, usually point mutations, single base pair changes, etc. Transposons, on the other hand, are big pieces of DNA that are moving around, jumping around all the time, and if anything, are probably causing at least as much mutational pressure in terms of changing the genetic information as any of these other kinds of mutation, repair, damage, etc. that we've talked about before. There are basically three main classes of transposons, and again, incredibly creatively named: DNA-only transposons, retroviral-like retrotransposons, and non-retroviral retrotransposons. The DNA-only transposons are really quite common in bacteria, and the main issue that we as humans have with these is very often they're also carrying antibiotic resistance genes. And so a lot of probably heard about MRSA, multi-drug resistant Staph aureus. The reason it's multi-drug resistant is because it's got a bunch of transposons in its genome, all of which are encoding antibiotic resistance genes. And these things jump around, not just in one particular organism. There are ways that we're not going to talk about, but that they can jump between organisms. So it's a big issue. Retroviral-like retrotransposons, these are probably just defective viruses, and that's probably the best way to think about them. And then these non-retroviral retrotransposons, again, negative, negative, negative kinds of things here. But these are the most common in our genome. About almost half of our genome is made up of these things, which is really kind of incredible. Quite why that is, nobody really has a good handle on it. But there are a bunch of them there, and can very often be causing mutations, causing changes in the genetic material. So how do you tell the difference between these things? Okay, yes, it's a retroviral-like or a non-retroviral retrotransposon. All of these are going to be DNA because they're in the genome. All of our genomes are DNA. How do you tell the difference? It has to do with these ends. And so these little arrows that you have here at the ends, these are indicating direct repeats of DNA if the arrows are pointing in the same direction, and inverted repeats if they're pointing in opposite directions. And we'll look at that in just a second on the next slide. Direct and inverted repeats are really critical in terms of thinking about transposons. So inverted repeats at either end are the DNA-only transposons. Retroviral-like retrotransposons have long direct repeats. You see the arrows in the same direction here. and that's because of how they replicate that we'll talk about before. And these non-retroviral retrotransposons are really kind of interesting because they don't have any of these kinds of repeats that are associated with them. You just see many copies of these kinds of retrotransposons inside the cell. So direct and inverted repeats. I get this question all the time. You'll probably see questions on the old exams that look like this. How do you know if it's a direct repeat or an inverted repeat? Best thing to do is to draw out the sequence, but not just with one strand, draw it out with both strands, and flip one upside down over here. So this is your double-stranded piece of DNA, 5' to 3', antiparallel, 5' to 3'. So a direct repeat is the same sequence on the same strand in the same direction. So here we've got GAAT, GCCA, N, what does N stand for? Any nucleotide. Molecular biologist shorthand for could be any of the four, followed by GAA, TGCCAT. So that's a direct repeat. Now, the opposite, as it were, of this is the inverted repeat, whereby you have a sequence going from 5' to 3' on one strand, and then that same sequence on the opposite strand, but exactly the same sequence again from 5' to 3'. So here, GAA, TGCCA, CAT, sorry, opposite strand, GAA, TGCCAT. So direct repeats, inverted repeats. Again, we'll talk a lot more about this as we move on, also talking about DNA binding proteins and sort of the reasoning behind why some of these have these direct and inverted repeats has to do with interactions of proteins, not surprisingly, sitting down in the major groove of your DNA. Yeah?
So the difference between these is how they're going to be read, just by their very nature, direct or inverted repeats, how they're going to be interpreted by proteins?
Okay, so basically the question is, what's the big difference between these two, direct versus the inverted repeats? Has a lot to do with, and what we're going to be talking about today is how the proteins are interacting with them are going to be sort of facing each other or head to tail, because that's going to be determined which orientation you have in your DNA, but also in terms of what happens when you have transposons that jump into or out of your DNA. And we'll see why that is in just a second. So two different things here. Why should we care? Hopefully still beating the dead horse here. Half of our genome, almost, are these kinds of elements. The excuse me, retroviral, retrotransposons, the line elements and the sign elements, the clearly retroviral-like elements, 8% of our genome, and then, of course, the piddly little protein coding genes over here down at the end. So way more of these kinds of things in our genome than anything else. But it's also true in bacteria. Even though bacteria have pretty streamlined genomes, there's often quite a lot of these transposons that are present in these particular genomes. And our favorite genome in Archaea, yet another group, is loaded with these things, which actually turns out to be a real pain when you're trying to sequence the genome, but that's a different story entirely. So what do these transposons look like in bacteria? Again, the main issue here, as far as we're concerned from a medical point of view, are all of these antibiotic resistance genes that are very often carried in these transposons. But it doesn't have to be carrying one of these antibiotic resistance genes. All of these transposons have to have is a transposase gene, and we'll look at what that transposase gene does a little bit later on, and then sequences at either end. Now, these are the inverted repeat sequences. They don't have the arrows here, but those are those sequences that are present at either end of these transposable elements. These jump around. How do they jump around? Well, there are a couple of different ways, but probably the most common and the most straightforward to think about are these so-called cut and paste transposons, which are exactly functioning like you would expect. They cut this transposon out of the DNA in one place, move it over somewhere else, and pop it in there. And so that's the process. What does that? It's the transposase enzyme. And here we have those nice little blue and yellow transposase monomers here actually come together as a multimer. They will bind to these inverted repeat sequences at either end of the transposon. These will then interact with each other through protein-protein interactions and make this wonderful zome complex. Again, these molecular biologists love their zomes. Protein nucleic acid complex. And once these come together, this literally will cut this transposon out of the DNA and move it into another piece somewhere else in the genome. When it does this, it cuts it out. It gives you a double-stranded break. That double-stranded break has to be repaired somehow. That's usually repaired through homologous recombination. But if you had, and there's actually a small amount of non-homologous end joining that happens in bacteria, it would get hooked back together here. But usually this is going to be a homologous recombination event which puts this back together. So now you have this transpososome, it's the transposase plus the transposon. It floats around inside your cell, finds a new place, it cuts the place that it's going to be putting itself into, and then integrates this transposon in the process. It's all the transposase enzyme that does that. This transposase enzyme is actually quite interesting. Almost all of these transposase enzymes, they don't cut the DNA immediately across from each other, so it's not exactly one base pair. It's almost always offset by a couple of bases on one strand versus the other. And that's supposed to be shown down here at the bottom. You see this little black stretch right here, black stretch here, and a black stretch over there. That's what happens when you cut these double-stranded DNA, offset a little bit, and pull those pieces apart. So you do that, you actually will going to generate a gap in your DNA sequence, but it's got a 3' OH, it's got a nice template. That will get extended and filled in by the normal DNA polymerase which is present inside the cell. But in that process, because this sequence here is the same as this sequence here, but on the opposite strand, you end up with a direct repeat just outside your transposon. So it's a directly repeated sequence, but the inverted repeat is actually in the transposon sequence, is what the transposase is going to bind to move that transposon along. So if you're looking at a genome sequence, you have direct repeat, inverted repeat, transposon, inverted repeat, direct repeat. That's a really good indication that you've now got a cut and paste DNA transposon. And if you look at bacterial genomes, there are lots and lots of these things spread around in. This is what this actually looks like at a higher resolution. Here is your blue transposase and yellow transposase subunit, which then bind to this transposon at these inverted repeat sequences. And inverted repeat just means, again, that these proteins are now facing each other, and will cut and religate and put these into the genome, leaving a little gap that gets filled in the next step. So DNA only, cut and paste transposons. Now let's talk about the most interesting ones. Oh, yeah.
Is this transposase interaction only with the inverted sequences or with the direct sequences?
So the question is the interaction of the transposase with just the transposon, so the inverted repeats, which actually are shown here in the red, or is it also interacting with and binding specifically, which is probably the more important point, to the target sequence? And the answer is it's not binding specifically to the target sequence at all. It's just when this interacts with the DNA, it now has a nuclease activity over here that's going to cut. So it's a separate piece, but it's not a sequence-specific interaction. And it's the direct repeat which is generated just because of those cut sequences. And if you think about it, it makes sense because this subunit is in a different place than this subunit. So they are going to be separated relative to each other when you come down and bind the DNA that you're going to be putting the transposon into. Yeah? Ian.
Can a transposonal element be part of a gene, just a segment, or is it always, or is it typically going to be an entire gene? Is it going to code for the complete protein, or can it just be any segment?
Okay, so the the question here basically has to do with definition of a transposon, and I'll just go for it that way. Basically, do you have to have extra genes that are with that transposon that are being moved along? Basically, in this yellow piece here, does this have to have a gene in it or not? And the answer is, all you really need are these sequences at either end, because these repeats, because that's what your transposase is interacting with. So if you happen to have transposase protein around, it can move those inverted repeat sequences and whatever's between them all around the genome. And that turns out to be exactly what happens in the sign elements that we'll get to in just a second. Okay, so retroviruses. This is total oversimplification. If you're interested in retroviruses, take my course next term. We'll spend a couple of lectures talking about retroviruses. Absolutely amazing from a molecular point of view. But the basic message here is that these are RNA viruses, so they have an RNA genome, which tells you they're totally cool anyway, because all the stuff we talked about is DNA genome so far. And that RNA, through the activity of reverse transcriptase, takes this RNA copy and makes a double-stranded DNA copy of that RNA. And then other proteins that are present in this retroviral genome will now take that DNA and actually in a manner really similar to the transposases, pop it into somewhere in the genome. That then will get made into RNA. That RNA gets put into a virus, and it goes off and does its own thing. But again, we'll spend a whole lecture talking about the mechanisms, and then other lectures talking about the individual viruses a little bit later on. But the key to this is really the reverse transcriptase. Now, everyone recognizes exactly what this structure looks like, right? What? Thumb, fingers, hand. Does that start to sound familiar? Like a DNA polymerase. It actually is extremely similar to DNA polymerase in structure. So yes, it takes RNA and makes a DNA copy from it, but it also takes that DNA copy that's made and makes another DNA from it. So it's actually not terribly surprising. This is not making RNA, it's just making DNA. So it's a DNA polymerase. It also has this extra little piece hanging off the bottom in red, which is, what is it called? RNase H. What does RNase H do? Chops up RNA when it's in an RNA-DNA hybrid. What's happening with this reverse transcriptase? It makes a DNA copy from RNA, but it's got to get rid of that RNA because it needs to make another DNA copy. So again, it's not surprising that it's got this RNase H activity as well that's associated with it. And that's just diagrammed right here. And again, all kinds of really cool stuff that we'll talk about next year in more detail. But then actually once you've made this double-stranded DNA and turns out put these direct repeats at either ends of it, how that works again is something we'll talk about next term. You've got a piece of DNA that the viral integrase can now interact with, and the integrase is acting very similar to the transposase we talked about with these DNA-only transposon. It binds to this viral DNA, chops the ends a little bit off the end of the viral DNA. Again, we'll talk more about that later. That then will move around inside the cell, particularly inside the nucleus, because these retroviruses almost all are infecting eukaryotic cells, and will cut your target chromosome with these little gaps at either end and insert the viral genome into here. So again, you've got short direct repeats at either end, but the big difference here is that instead of having inverted repeats at either end of the genome, these are now direct repeats relative to each other. And so if you have short direct repeat, which is due to the cutting and refilling, and then a long direct repeat, short direct repeat, you now know that it's a retroviral integration as opposed to a DNA-only integration. So again, we'll talk a lot more about the details in virology next term. 421, 521, sign up for it, great class. The other kind of retroelement are these non-retroviral retroelements. Now, why is it called a retroelement? The reason it's called a retro element is because just like with reverse transcription, it has an RNA intermediate. And that RNA intermediate is absolutely required for getting the replication of these things, but also inserting them into the genome. So what happens? You have one of these elements that's already in your genome, a little bit hard to see here, but it has it's a repeated sequence, also encodes proteins, as long as it's a line element. This is particular one is line element 1. Line, by the way, I should have put this down here, stands for long interspersed nuclear element, which means you've just got, they're long, there are a whole bunch of them all over the place. Signs are, what's S stand for? Short. Interspersed nuclear element. So again, many copies of these. They all have, and this is the sort of defining feature of these, a stretch of A's on one strand, and of course that means there's a stretch of T's on the other strand. And that should be kind of an automatic giveaway that this was made as a messenger RNA. Messenger RNAs have these poly A tails that get added to them. We'll talk about those probably next week, how that actually happens. And those poly A tails serve as the template for a reverse transcriptase, which in these line elements is encoded in that particular transposon, getting to your question about what genes are actually there. So that reverse transcriptase will make a DNA copy of this, but like all DNA polymerases, they all need primers. Where does that primer come from? Turns out the primer is actually the DNA that is where that retrotransposon is going to go into. So this gets cut by another protein, which is present in these retrotransposons, an endonuclease, which now provides a 3' OH. Now the reverse transcriptase can copy the RNA, and just like the case with the retroviral reverse transcriptases, can also make the opposite strand. This now can get integrated into the genome, and literally about 40% of our genome is made up of these kinds of elements. They seem to be ridiculously successful, and it's true for almost all vertebrate genomes as well. You've got very, very large numbers of these, and it's really dependent on having these reverse transcriptase endonucleases, which can cut pretty randomly in the genome and then make copies of these. And getting back to your question about are these things encoding that, the long interspersed nuclear elements are encoding the endonuclease reverse transcriptase. The short ones aren't. And they have sequences in those particular things, which are basically targets, like you have the inverted repeats at the end of those DNA-only transposons. I'm not ignoring you. Question, yeah.
There's a difference between reverse transcriptase and the retro element replication is that the DNA conversion is happening on the DNA strand as opposed to being built and then inserted into the genome?
Yeah, that is exactly correct, and I'll just repeat it. The big difference between these two is that with the viral-like reverse transcription, that double-stranded DNA is made by the virus, or in the case of these retrotransposons that are like retroviruses, made in the nucleus or in the cytoplasm. This is a separate piece of DNA that then gets inserted, a lot like what happens with the DNA cut and paste. Whereas in this case, there's no extra DNA that's not already attached to the genome. So the DNA is actually made in situ from that RNA. Yeah, it's exactly correct. Okay. More questions about transposons? And if he does something like that, what does he mean? Time to get your clickers out. So, which of the following types of transposable elements are most common in the human genome? Oh, did I not start this? Sorry. Start. DNA-only transposons, IS elements, cut and paste transposons, retrovirus-like retrotransposons. And why is this not doing what it's supposed to do? Sorry, it's not counting up here. Oh, it is actually counting up. I'm just not sure what the time is doing. Okay, I'll just tell you what the time is. 40. 30. 10. 5, 4, 3, 2, 1. Okay, let's see if I can actually move this thing now. Yes, the right answer is E. Why my mouse is not doing this? Here we go. And most of you agree. And sometimes I wonder with these clicker questions, it's just a short-term memory thing, seeing if you're taking notes and being awake. Never quite sure what the process is here. So any questions about this? Turns out that if this were a question about bacteria, it would be the DNA-only transposons. IS elements I didn't talk about, but actually an IS element is exactly what you were asking about before. It's one of these direct repeat sequences without a transposase that's associated with it. Cut and paste transposons are actually these DNA-only transposons. Retrovirus-like, about 8% of the genome, but about 35% of these non-retroviral elements. Yeah?
So how would the IS element be different than a sign?
So the IS element is now one of those DNA-only transposons, as opposed to the sign, which is a retrotransposon. So it's going through that different step that we talked about at the end. Okay, other questions on these kinds of transposons? Okay, so I wanted to spend a couple of minutes talking about site-specific recombination. This is now, instead of the random or more or less random kinds of integrations that you get with the retroviral, the non-retroviral retrotransposons, and the DNA-only transposons, this is now where you have a very specific sequence in the genome that you now have the integrase, like the very similar proteins to your transposases, that are bind to a very specific place in the genome and put whatever it is that they have with them into that particular position. And there are a couple of interesting things about this, and it has to do with direct repeats versus indirect repeats. So it turns out if you have a directly repeated sequence, like you have here, between whatever it is that you're interested in with your site-specific recombinase, and that's a direct repeat, so it's exactly the same sequence, 5' to 3', in that sequence and your genome, in the presence of a recombinase, which basically is going to take one of these, cross it over, and hook these two ends together. We'll take a look at that in just a second. That will integrate this, whatever piece it is, into the genome with these direct repeat sequences on either side. And this, just like all other chemical reactions, is a reversible reaction. So it can cut this piece back out, and this piece has been removed. If, however, you have inverted repeat sequences on either end of this, taking one end and crossing it over with the other just flips this piece of DNA around. So it's what's called an inversion event. And it turns out that both of these happen. They're both dependent on a protein which can cut one of these strands and hook it over to the other one. The best understood of these, of course, is viruses, because viruses are the coolest thing ever. Bacteriophage lambda has been really the best studied of any of these. It has a circular chromosome once it comes inside the cell. Again, whole lecture next term, by 421, 521. We have a directly repeated sequence in the viral genome and a sequence in the host genome, which is completely identical to it. There's a viral integrase protein, the lambda integrase protein, which now binds to this sequence and links the two of them together, giving you this viral gene copy in the genome. This integrase can also back up, chop the virus sequence back out, and make lots of virus. Virus life cycle overview, again, talk a lot more about it next term, where you have a lambda virus injects its genome inside the cell. That becomes a circular genome, and we're just going to talk about this side over here. That circular genome with the lambda integrase gets popped into the genome, and this is great from the virus's point of view, again, to totally over-anthropomorphize here, because now every time the cell divides, the virus divides too. Its genome is present. It just continues to be maintained. That's probably most of the viruses in the environment replicate through this kind of mechanism, because it's great if the cell does all the work for you. You don't have to bother with this, annoying infection business and finding other cells, etc. However, there are cases where this is actually not such a good thing, and it turns out this is almost always because of DNA damage. So DNA damage happens to the host cell genome, and that actually gets sensed by the virus, and the virus says, hey, I'm DNA, I'm in this genome, I need to get the heck out of here because my DNA might be being damaged. And so that process, that again, we're not going to talk about here too much, leads to this virus genome getting chopped back out and then eventually making a whole bunch of virus particles. But as far as we're concerned, for this class anyway, it's an identical sequence that you have in both the virus and the host, which once it integrates gives you a direct repeat. That can then be chopped back out, which will give you a separate circular piece. Or if you have, again, these opposite orientations, you can have flipping that happens of different genes. And it turns out, again, when we talk about gene regulation a little bit later on, this does turn out to be extremely useful for some kinds of bacteria. Another reason that this is a really useful technique is not so much from a biological point of view directly, but it's a really, really useful research tool. And what happens in using this research tool is basically what you do, again, we'll talk about this right at the end of the class, is you can take a particular gene and put recombination sites, these direct repeats on either end of that particular gene. And you do this through cool molecular biology techniques, again, that we'll talk about at the end of the term, or take that theory part of the recombinant DNA stuff, 430 next term. But you have these direct repeats on either end of the gene that you're interested in, and that in and of itself is actually quite stable unless you have one of these integrase recombinases around. And what you can do, and people do this all the time, is they have this recombinase gene that they will make, but only in very specific cells at very specific times, usually in development. And when this gene is expressed, then you will have chopped out whatever this gene is here, and this circular piece of DNA doesn't have a replication origin, has no way of being segregated, so this gene is actually lost in this process in the presence of this recombinase gene. And you can design, usually this is done with mice, a situation where this particular recombinase gene is only made in specific places at specific times. We'll talk about how that's regulated a little bit later on. But for instance, here in the liver. In the liver, the Cre recombinase gene is made. The binding sites for this recombinase gene are called lock sites for all kinds of historical reasons that we won't get into. And if that's the case, this gene gets chopped out. On the other hand, in a lot of other places, because of this regulatory region, this recombinase gene is never expressed. So your gene is perfectly happy. Now, why is this something which is important? If you want to study a gene, say for instance in mice, that is absolutely required for that mouse to function, say a brain development gene or something like that, you're interested in what that gene is doing in a different part of the mouse, doing this process allows you to have very specific gene deletion at particular times and places inside a whole organism. And what this is called floxing. The molecular biologists love to talk about flox. I floxed to this gene. What is floxing? Floxing is putting two flanking on the outside of your gene lock sites, and then when this Cre recombinase is being expressed, it will chop this gene out. Sometimes you'll put this into the genome. Often what people do is actually put this gene onto a virus. And that virus then, when it has the infection, will encode this gene, which will then cause any of the cells that are infected by that virus to lose whatever gene it is that you're looking at here. So it's a really powerful molecular biology technique for dealing with causing gene deletions. And it's all because people were studying how boring bacterial viruses worked, i.e. lambda. And Cre turns out to be extremely similar to lambda. So that's what I want to talk about in terms of jumping genes. We got our DNA-only transposons, again, mostly present in bacteria. Really problematic because many of them, not all of them, but many of them carry these antibiotic resistance genes. Retroviruses have the RNA, which gets made, thank you, outside of your actual genome, and then gets integrated through a mechanism extremely similar through these DNA-only transposons. And then the other retrotransposons are the ones which will hook up to the DNA and make a RNA into DNA, but when it's actually attached to the place that it's going to be integrated. And these are the ones that are extremely common in our genome. And it turns out also cause a number of mutations. A lot of the spontaneous hemophilia, which happens from one generation to another, is actually due to these non-retroviral retrotransposons jumping. And so there's some really nice examples of exactly how that works. And people can actually literally see these moving around in humans, and particularly people who've come down with some of these genetic diseases. Site-specific recombination, we know about it mostly through the study of lambda bacteriophage, but it turns out to be a really useful tool for doing, they call it recombineering. Again, a horrible word, they love to make these things up, but it's recombination for genome engineering and using what we know about that. So, talked about homologous recombination the last time. Any more questions on homologous recombination? No. Why is it happening? Mostly due to double-stranded breaks, but also in meiosis. You can get crossing over that takes place. Mechanism, your 3' extension gets made, coated with these recombination proteins, strand invasion. Yeah.
It says that it's not typically used in humans outside of meiosis.
Homologous recombination is not typically used in humans. It says, that would be the textbook says. So it really seems to vary. So not commonly used in humans. Non-homologous end joining, the quick and dirty method, is probably used more frequently in most cells most of the time. But there certainly are cases where you do have homologous recombination that takes place. And the fact that when you have mutations in some of those homologous recombination genes, you end up with a lot of cancer cases as well, which probably aren't just due to meiosis, because you're seeing these cancers forming in cells that have nothing to do with the germline. So yes, probably more common that you've got non-homologous end joining, but certainly not only under those kinds of cases. Hybridization, that's your strand invasion. These REC-A, RAD51 proteins, this is binding to that single-stranded piece. And then holiday junctions, hopefully some of you had a chance to look at the clemations. I got a question from someone about exactly which ones are which, but I find that those are really useful in terms of thinking about how these things interact with each other. Yeah?
Just to be clear on what a heteroduplex is,
Mm-hmm.
that's two strands of different DNA, like
Non-complementary. Yeah, so the question basically is what's heteroduplex DNA? Literally, heteroduplexes come from two different places. So that's what it is. It could actually be completely complementary sequence, or it could have some differences between the two. And if you have differences, then you can end up with things like mismatch repair that ends up leading to gene conversion. But heteroduplex just by itself just means it's come from two different sources. And again, which can be different, but it not necessarily so. Okay, and then with site-specific recombination, we talked about that, and then our various different transposons. So we're done with recombination until we finish our last clicker question. Integration by bacteriophage lambda is most similar to integration by DNA-only transposons, Cre-recombinase, replicative DNA transposons, retrovirus-like retrotransposons, non-retroviral-like retrotransposons. And again, feel free to chat about this. $20 seconds. 15, 10, 5, 2, 1. Okay, we seem to have some differences of opinion here. So integration by bacteriophage lambda is what? It's a very specific kind of integration. Is it DNA or a DNA copy that you've gotten from RNA? It's just DNA. So we have our replicative DNA transposons, retrovirus-like retrotransposons, non-retroviral-like retrotransposons. We can get rid of these retrotransposons because it has nothing to do with RNA at all. It's just DNA. So that brings us to our last three. DNA-only transposons, Cre-recombinase, replicative DNA transposons, etc. Cre-recombinase, actually, as it turns out, is not part of a virus. So it's not actually an integration. So B, it can't be right. So we're down to A and C, replicative DNA transposons or DNA-only transposons. Replicative DNA transposons I didn't even talk about. So it might be the same, turns out it's actually quite different. So that actually only leaves us A. Okay, so now, change subjects. I want to talk about transcription. This is just the outline of what we're going to talk about today. We may or may not get all the way down to here. Probably not going to get actually past eukaryotic transcriptional initiation here. Transcription, this is exactly the same thing that we had very much at the beginning. It's all about going from DNA to RNA. And by definition, this is transcription. So we'll talk a little bit about the RNA, mostly talk about the RNA polymerase, and I should be more specific here. This is the DNA-dependent RNA polymerase. So it takes a DNA template and makes RNA from it. Now, it turns out where you start, actually not unlike the case of your origins of replication that we had for DNA, is really the most important thing in terms of what pieces of DNA get made into RNA as opposed to anything else. And unlike the case with DNA replication, where you need to make the whole genome, now you just need to make RNA from a relatively small part of the genome. And so that's why initiation is really critical. So if you just look at a genome, turns out that looking at where RNA is really kind of hard. Sequencing DNA is really, really easy. So one of the big questions that's come out of this whole genomics is, what is a gene? And what is being transcribed? And can you figure that out just by looking at the DNA sequence? And the answer is, it's kind of hard, actually, to be able to do that. So there are a couple of different ways about going about doing this. So this is an example from the human X chromosome. Only 155 million base pairs, which is one of the larger chromosomes. We're looking at a very, very tiny piece of that particular genome here. Just as we've looked at before, all of these, the horizontal line here is just your DNA. and anything which is dark is an intron that gets cut out. We'll talk about cutting out a little bit later on. Is these thin vertical lines are the things which are actually coding for proteins. So that we only know actually by looking at the proteins later on. It's kind of hard to tell what these things actually are. How do we know, if we don't know what the protein is, that these are likely to be important sequences? Well, one of the ways to know is to use conservations. This goes all the way back to lecture 1, talking about things that are generally important are likely to be similar between particularly similar organisms. Things that are important on the completely global scale are going to be conserved all the way across all of these different organisms. In this particular case, we're looking at vertebrate genomes, which are all relatively similar to each other at a molecular level. But one of the interesting things you see here, if you look at the 100% conservation in all these vertebrate genomes, you get a tall line here. That means that they're all relatively well conserved. And turns out that these match really well with protein coding sequences. Is that you see them all the vertebrate genomes, they're all here. In these areas where there aren't any kinds of genes, they're not very well conserved. So it's actually a really great way of figuring out that there's something interesting there. However, this is not perfect by any stretch of the imagination. If you look down here, there's a bunch of conserved sequences. They're in this intron. A couple of conserved sequences here in this piece of DNA that we have no idea what it's actually doing. So it's a good start, but it's not going to tell you something completely definitive. There are a few other ways of looking at this. Non-synonymous substitutions. It's a non-synonymous substitution. We'll talk more about this when we talk about translation, particularly next Friday when the dean will be here. Puzzle spot the dean when she gets here. The difference, non-synonymous, means a nucleotide change between two different organisms that causes a difference in amino acids once you actually have that. So if you have a particular position here, like this triangle, it's a non-synonymous difference between humans and Neanderthals. We only got the Neanderthal genome sequence about 4 years ago. So it's really pretty amazing that you can look at some of these things. But if you look where these are, they're all in very specific protein coding sequences. And it turns out to be important genes that are probably giving us differences between Neanderthals and modern humans, and actually tells us a lot about the evolution that just happened in the human lineage here. So the other thing which is probably totally bad on here, yes, we have these sequences from Neanderthal about 4 years ago. All these purple triangles here, we should probably have about 10 times as many of them on here. These are all of the small RNAs which are present in this particular part of the sequence. And actually, there are probably a lot more which are in there. We don't know what all of them are. So, take-home message from here. Yeah, genes are all over the place. Somebody asked, I forget who it was, last time about, if you have these kinds of interactions, like the breakages, like we're talking about non-homologous end joining, are genes more likely to be mutated or less likely to be mutated? Are they packed? Are they less packed? This is a pretty good example that they're spread all over the place. And that's actually a pretty gene-dense part of the X chromosome, which is here. So there's no rhyme or reason to that. Homology is variable, but finding homologous positions is usually going to be very important for something which is going on in that sequence. It could be protein coding, it could be RNA coding, or it could actually be some kind of regulatory sequence which is present there as well. And a lot of those regulatory sequences are going to be promoters, which are your initiation point relative to getting transcription to take place. So what gets made, it's the RNA. Again, this is a repeat from what we had before. It's the ribose sugar as opposed to the deoxyribose sugar. You've got uracil instead of thymidine. And you can have all kinds of extra funky base pairs. And some of my favorites are actually shown down here at the bottom, where you've got these bases reaching out here over to another totally different piece of RNA over here. Ends up forming sort of a triple base over here. So lots of non-standard base pairs. When I say non-standard base pairs, these are the non-Watson and Crick, AUGC kinds of base pairs. And of course, when Stedman puts quotes around something, that means it's not really single-stranded. Most of your, RNAs actually look more like this down here at the bottom, all wrapped around each other, base paired, normal base pairing, non-normal base pairing, etc. But again, other than the uracil and that extra OH that we have here on the ribose, as far as polymerization is concerned, as far as the chemistry is concerned of all of this, it's extremely similar between DNA and RNA. Difference on the base is just this one methyl group right here on the thymine. So extremely similar to each other, and in terms of all of the activation, the actual chemical reactions, pyrophosphatase formation, nucleotide triphosphates getting cleaved, all of this stuff is absolutely identical between DNA polymerization and RNA polymerization. Also always goes 5' to 3', etc. So what kinds of RNAs do you find inside the cell? Most people talk about messenger RNAs. This is the ones that are going to cause the proteins. These are the ones which are all those exons that we saw in our genome map right at the beginning. Turns out actually not very many of these. Only about 3 to 5% of the RNAs inside a cell are these messenger RNAs. The vast majority of RNA inside a cell is ribosomal RNA. And again, when we talk about translation, we'll see that the ribosome is almost completely RNA. So if you just purify RNAs from cells, it's mostly ribosomal RNA. Actually, in second place are these tRNAs, and then messenger RNAs are relatively infrequent. It's actually kind of interesting. It took a long time for people to discover messenger RNAs because there weren't very many of them inside the cell. And then even later were all of these other RNAs. And all of these other RNAs down here, various different abbreviations, etc. We're not going to talk about most of them. We will talk about these snRNAs, snORNAs in more detail. These are important actually for modifying other RNAs. SNO-RNAs in particular are important for ribosomal RNA modification. Small nuclear RNAs are most important for splicing. And then we've got lots of different regulatory RNAs, and we really don't understand what most of these things are doing. And there's huge numbers of them here. Huge numbers. Sorry. Had to. There are many, many more of these that are around, and we really don't know what most of them are doing. We had a little bit of a discussion about some of these long non-coding RNAs. The telomerase RNA is a really nice example of that. And then our candidate, actually, if anybody was at that, I think only one or two of you came to that seminar last time. I'll try and remind you, next week, Thursday, is going to be a new job candidate coming in. We'd love to get feedback from undergrads in terms of those people here. But he's also working on these long non-coding RNAs. So why are RNAs so useful? One of the reasons that RNAs are so useful is that they can take particular parts of the genome and make many copies of that particular part of the genome. And so this is the amplification process. And I mentioned this again when we were back at the beginning. You can have one particular stretch of DNA, say this particular gene. If you get a lot of transcription from that gene, you're going to end up with a lot of the protein that particular gene is encoded. Whereas if you have a very small amount of transcription of this other gene over here, you're going to have a lot less of that protein. Again, when we talk about translation, it turns out that each of these RNAs can be made into large amounts or smaller amounts of protein. But for this purpose, we're just going to talk about the transcription, how much RNA is being made. Almost always more than just one copy of whatever is being made there. What does this job? It's the DNA-dependent RNA polymerase. The DNA-dependent RNA polymerase will copy one of the strands of your DNA. That's your template strand of DNA. And again, 5' to 3', just like the DNA polymerases, but one really big difference is that these DNA-dependent RNA polymerases do not need primers. They can start by themselves. And this is one of the very general cases of all the RNA polymerases we're going to talk about in this particular class. You remember the DNA primase? What's DNA primase? It's an RNA polymerase. Why is it so important that it's an RNA polymerase? Because it doesn't need a primer. Because it's making those primers that all the DNA polymerases can actually extend from. Interestingly enough, turns out that these DNA-dependent RNA polymerases have much lower fidelity than the normal DNA-dependent DNA polymerases, all the replicative ones, mostly because they don't do the same kind of proofreading. There's a little bit of proofreading that happens in these polymerases, but it's a lot less than what you see in all of the DNA polymerases. The RNA, once it's been made from this template, actually is rapidly released, i.e. that means it doesn't stay in this DNA-RNA hybrid for very long. Actually is removed quite quickly. We know a lot about the structure of these DNA-dependent RNA polymerases, and again, this is something which has happened literally in the last 10 years. Almost every other week, there's a new structure of some other part of the DNA-dependent RNA polymerases, which is being solved and people are figuring out. Dr. Mike Bartlett here, who teaches the recombinant DNA techniques theory section, his lab actually works on these particular DNA-dependent RNA polymerases. Very interesting kinds of structures. They are also relatively slow, 20 nucleotides per second. That's compared to the replicative DNA polymerases, some of which can be 1,000 nucleotides a second. So it's a much slower process, which actually happens here. So let's look at one of these processes on how you get transcription taking place. Transcription has to start somewhere. What does that start? It's the promoter. And the promoter is true for bacterial transcription, for eukaryotic transcription, for archaeal transcription. It's where the DNA-dependent RNA polymerase interacts with the DNA. It's where it's going to start. Then you have this process where you have the DNA-dependent RNA polymerase that binds to the promoter. And then what it has to do, because it's got to have a template, what it needs to do is pull the two strands of DNA apart. So it's basically a helicase activity. But instead of having the helicase activity separate, it's a separate protein, which is what you have with DNA polymerization. Here, it's the RNA polymerase itself that's pulling those two strands apart. That's also called the open complex, open because you've pulled the two DNA strands apart. After you have open complex formation, then you have something called abortive initiation. And the reason that you have abortive initiation seems to be that these DNA-dependent RNA polymerases, again, in a difference from the DNA-dependent DNA polymerases, are insanely processive. Once they get on and they start moving, they will continue to go until they get to a termination site. So it's not like the DNA polymerases that are popping on and popping off unless they have a separate sliding clamp to keep them on. These are incredibly processive, and in some cases, actually, I think there's like certainly hundreds of thousands of nucleotides that some of these RNA polymerases can make all at one time. Because, particularly if you've got a promoter that is binding to a lot of these polymerases, you could end up spending huge amounts of energy. Remember, it's the same chemistry that we have with the DNA polymerizations, nucleotide triphosphates. So if you undergo lots and lots of transcription at a gene that you really don't want or don't need, it's a huge waste of energy. So there's this process called abortive initiation, which is basically sort of the DNA-dependent RNA polymerase kind of spinning its wheels for a little while until it gets traction and takes off. And that's, again, probably an energy conservation mechanism because of this really high processivity. Once you get going, you're going to burn a lot of nucleotide triphosphates. And so you really want to be sure that this is something that you're going to be extending. Yeah?
So during abortive initiation, is that like a checking time where it's
So basically, the question here is that kind of a regulatory mechanism. It's not really a proofreading mechanism per se, because that would be actually changing the nucleotides. But it is a way of just sort of, again, controlling whether this gene is going to be transcribed or not. And what this does, and when we talk about gene regulation later on, it turns out that this is a place that you can regulate transcription. You either have more of the abortive initiation or less of the abortive initiation. And we'll talk about how that gets impacted on that process. Once ever that decision gets made, that you do want to, the wheels have stopped spinning and you got traction, this takes off. And one of the things that happens is there's this big conformational change that happens in the protein. And so basically what it does is grabs onto the DNA, not unlike the sliding clamp, but now it's part of the RNA polymerase. And that's partly how it stays on the DNA until it gets to a specific place. And this is usually, again, it's a specific DNA sequence that the RNA polymerase gets to, and then termination happens and it falls off. But it has to be a very specific kind of sequence and a very specific kind of mechanism. This whole process, closed complex formation, open complex formation, abortive initiation, elongation, and termination, is general for everything. You probably notice I haven't said bacterial transcriptions yet. Now I'm going to talk about some of the very specific differences that you have between bacterial and eukaryotic transcriptional initiation. The first one is this sigma factor. So sigma factor is part of the RNA polymerase. When people say factors, all these molecular biologists say factors, they just mean a protein. So sigma factor is another protein that associates with the DNA-dependent RNA polymerase and gives you specificity. It says, hey, this is a place on your DNA that you want to bind to. And so it's that sigma factor which says, okay, this is the promoter. This is where the polymerase is going to bind to. Once you've undergone abortive initiation and actually changed to go to the elongating mode, this sigma factor is actually released, and that's one of the big conformational changes that happens in the bacterial polymerase that allows this elongation to take place. And this sigma factor can only rebind to the polymerase once it's terminated and it's been separated from the DNA and is ready to restart again. Also, termination is different in bacteria, and we'll talk about that in a little bit more detail after we talk about promoters. So, the sequence that the bacterial DNA-dependent RNA polymerase can bind to, due to its sigma factor, is the promoter sequence, wherever you start. But that particular sequence in bacteria, again, is what's recognized by the sigma factor. So the sigma factor is going to recognize a particular sequence. And this is a particular sequence here. I don't expect you to remember the sequences here. But TT-GACA, TAT, AAT. So how do we know what these sequences are? One way is actually just to look at what sigma factor containing polymerase actually binds to, and then look at the sequence of that particular DNA. But it turns out that the DNA polymerase, the DNA-dependent RNA polymerase, can bind to lots of different sequences. It turns out that some of the sequences it binds to really well, and if it binds really well, that means that there's going to be a lot of RNA polymerase bound there, lots of open complexes which get formed, lots of transcription that takes places. And then there are also some sequences that the RNA polymerase is going to bind to, but not as well. And it turns out you can just look at, take the case of E. coli, which is what this is from. There are about 4,000 genes, give or take, in the E. coli genome. Most of those have a promoter that's associated with them. So you can take all of these promoters, and just like you did looking at sequences from different organisms and finding out which ones are likely to be important, you can also do this with all of the promoter sequences and line them up and see which ones are important. And one way of looking at that is this kind of plot that we have here. At this first position, which turns out to be 35 nucleotides away from where transition starts, most of the time you have T's, but sometimes you have the other nucleotides. Here, sometimes you have A's, sometimes the other nucleotides. Here, mostly T, A, A's, and T's. And one way of looking at that is just down here. The size of the letter represents how frequently you have that particular nucleotide at that particular position. So we'll start again here next time. We'll see you on Friday.